Hey guys, all right, find a seat. We still have a lot of croissant left. You guys are thirsty. Yeah, so first off, thanks to our sponsors. Raven just re-upped their subscription, or not subscription, but their sponsorship. We're always looking for more sponsors so we can buy more pizza for you guys next time. So if you want your face or your logo on here, just send us some cash. We'll put it up there for next time. Yeah, without further ado, James Roney, he's a graduate student here at MIT. Actually, I knew James since before MIT. Like we both actually met at Harvard over Zoom. Yeah, so it was over during the pandemic times, and so it's really exciting to see him progress now as a grad student. And some fun facts he told me. Let's see. Staring. Apparently, he never lost a staring contest. So if you guys want to challenge him after, and then I think he won a rap battle or a championship of rap battles in middle school. So that's also, you guys want to take him up on that?
Yeah, really, definitely setting myself up.
Yeah. All right. So afterwards, stay behind and challenge James. All right. Well, with that, I'll let James take it away.
Great. Thank you, Sergey, for the introduction. Yeah, I'm going to be leaving immediately after the talk, so running away from any challenges. So, yeah, thanks, Sergey, for the great introduction, and also thank you to Chris and Nick, and also Sergey for organizing this club. I look forward to these meetings every month, and I think I speak for a lot of us when I say that these meetings are a great source of food for thought and also actual food for hungry grad students. So yeah, so for those of you who don't know me, I'm James. I'm a second-year grad student in Sergey's lab. And today I'm going to talk about deep energy-based models for protein structure, dynamics, and folding. And so this project has been the focus of my PhD so far, and I hope that it serves as a starting point for a lot of interesting future work. Yeah, so basically there are a couple of elements of what I'm going to talk about. Basically, I'm going to start with some background on the protein folding problem, protein structure prediction, and AlphaFold. And then I'm going to talk a little bit about modeling protein energy landscapes. Then we'll go into the model that I developed, ProteinEBM, what it's capable of, how we can use it for protein design, because this is a protein design club. We'll talk, if there's time, I'll talk a little bit about model interpretability, and then talk about some of the future directions that I'm really excited about for this work. Okay, great. So as everyone, most people in this room probably know, energy landscape theory tells us that proteins are going to fold to a native structure that minimizes free energy, and we like to visualize this on a sort of reduced dimensionality surface like this, where we think about the polypeptide chain folding in such a way that satisfies all of its hydrogen bonds, buries hydrophobic residues, and trades off enthalpy and entropy to arrive at a folded structure. And so we, for a long time, have thought that, because sequence encodes structure through this energy landscape, we should be able to predict protein structures from protein sequences. And AlphaFold is this really exciting model that came out about 6 years ago now that seemingly is able to do this. So it takes in a protein sequence, a multiple sequence alignment, which is a set of related sequences from like other species. And then it processes these through a deep neural network, and then out comes a structure. And oftentimes these structures are very correct. So, so is that it? Did AlphaFold sort of solve the protein folding problem? Is it capable of sort of searching this energy landscape and finding minimum energy structures? So the, sorry, is this, so the short answer that I'm going to go to is no, because AlphaFold actually AlphaFold kind of cheats at this problem. And the way it cheats at this problem is it uses the rich signal that's present in multiple sequence alignments, this coevolutionary signal. So basically, if we compile a multiple sequence alignment, if we take a protein and we find homologs from a human, this little fish guy, this little cat guy, and this little starfish, we can analyze patterns of covariation inside this multiple sequence alignment. And what we sometimes see is that there are residues that seem to be correlated inside the sequence alignment. And what this can be indicative of is that they're actually in contact in the folded structure and sort of constraining each other in mutational space. So if you have one residue that mutates, then that can induce selective pressures for a compensatory mutation at the other contacting residue. And basically, if you look at these covariant contacts and overlay them with crystal structure contacts, they actually do a really good job predicting them. And there's a very rich body of work in turning this coevolutionary signal into a powerhouse of protein structure prediction. Lots and lots of people who have worked on it. Some of those contributions are summarized here at this nice website that Sergey has put together. And many of those people who have contributed to this body of work are actually members of the protein design and modeling community here in Boston. So this is a super powerful engine for protein structure prediction. And it's really the key to the success of models like AlphaFold. And we know that because if we take AlphaFold and we remove these evolutionary inputs, so we get rid of the MSA, and we just say, here's a single sequence, can you fold it into a folded structure? AlphaFold generally fails. So without multiple sequence alignments, AlphaFold takes in this single structure, it processes it, and it tries to produce a structure prediction. And so if we look at the distribution of how accurate those structure predictions are, they're much, much less accurate when you remove the multiple sequence alignment. When you have the multiple sequence alignment, they're all clustered here near the 1 TM score range, which means that they're accurate. And then with just single sequence, they're much less accurate. Below 0.5 TM score is generally regarded as a different fold, generally pretty inaccurate. And this is actually consequential. So for a lot of proteins that we're interested in, we do have deep MSAs and we're able to predict them well. But this is still a significant problem. Most protein design pipelines use AlphaFold as a final sort of verifier to fold sequences into structures and sort of see whether the designed sequence folds into the intended structure. And what we see here that AlphaFold actually isn't that good at folding protein structures from single sequences. So this kind of constrains the sequences that we're able to design to this narrower space of proteins, which we hypothesize are proteins that have simpler folds that are easier for AlphaFold to sort of solve in this single sequence mode. So that's a problem, and potentially being able to solve folding from single sequences would enable a broader range of designable proteins. And then also there are proteins without an evolutionary history that we care about, like viral proteins and various orphan proteins. So this is a problem. And so a long time ago, actually, this is work that I did many years ago when I met Sergey on Zoom back at Harvard. We were thinking about why this is the case. Like, why is AlphaFold struggling to predict these proteins from single sequences? And we came up with this idea that, well, maybe AlphaFold has learned something that's kind of akin to the free energy surface of a protein. But what's really the hard part is being able to search this vast space of possible conformations and find one that's in a low energy. This is something that's known as Levinthal's paradox in the protein biophysics community, and it's kind of asked this question of how are proteins supposed to explore this vast conformational space. And so we were thinking that even if AlphaFold has learned some accurate sort of internal scoring function that can recognize a good structure once it's found it, maybe the search problem is just too difficult. And that the MSA actually, by allowing AlphaFold to infer some of these contacts, can sort of warm start this search process. And then when AlphaFold then produces its output confidence metrics, those confidence metrics will sort of track, how well the prediction scores in this sort of learned potential function. And so to test this idea, basically, we took away the MSA from AlphaFold, and instead we replaced it with a template. So AlphaFold can take these template inputs, which are supposed to be solved structures of homologous proteins, and it can also use those in place of an MSA to sort of warm start its search for a structure prediction. But what we did was we actually had a bunch of templates that were of varying accuracies, some of which are decoys, so they have the same sequence as the true structure, but actually the structural elements are arranged wrong. So the helices are packed together, some of the secondary structure assignments are wrong. And we wanted to see, is AlphaFold going to get tricked by these things and spit out a high confidence prediction, or does AlphaFold sort of, is AlphaFold able to recognize a good candidate structure even in the absence of the MSA information? And so what we found was that when we gave these decoys to AlphaFold and we compared sort of a score based on AlphaFold's confidence metrics and the true accuracy of these templates that we were feeding in, these things were very well correlated. So what this means is that even though AlphaFold can't predict these protein structures from scratch, when it's given a correct structure, it actually is really good at recognizing that structure is correct, which we think supports this sort of hypothesis that that what's going on here is that the MSA helps sort of warm start this hard search problem of finding a low energy conformation. But AlphaFold's ability to sort of recognize correct folds extends beyond what you would maybe think from its poor performance at predicting proteins from single sequence. Yeah.
Yeah, this.
Yeah.
How much does this result depend on masking the sequence of the template?
Oh, yeah, that's interesting. We found that so this is a little bit technical, but AlphaFold has multiple sequence input tracks, one of which can be associated with the template input. We found that if you gave it sort of the native sequence as the template sequence, and that perfectly matches sort of the target sequence, which is this other input track, AlphaFold would sometimes be overconfident in even bad decoy inputs. So we found it to work better when we masked out that sequence. But since then, other people who have applied this method, particularly with protein binders, have found that it actually works better when you don't do that. So in our setting of monomer ranking, we found that that was important. But other people have since used a very similar method and found that it doesn't work as well. But I think that this is not a particularly consequential thing. Like it's a very technical sort of how the model is wired sort of thing. I think the important thing is that, well, I don't know. I mean, like, yeah, so I guess in this setting it was important, yes. But I wouldn't say that that's sort of like a blanket that applies everywhere. Yeah, so also we found that when we give these incorrect structures to AlphaFold, AlphaFold actually is generally able to improve them a little bit, but generally not all the way to a perfect structure, which again supports this idea that AlphaFold is sort of performing some sort of internal search process, and that we can sort of seed it with these structures of varying accuracy, and then the confidence metrics tell us sort of how well we've sort of descended this scoring potential surface. Yeah, so basically, to sum that up, AlphaFold does a great job at identifying candidate protein structures, even if it can't predict those structures from scratch. And it suggests that this global search problem of finding a minimum energy structure is really difficult with the amount of compute that sort of a neural network has inside of its layers. And so this sort of made us ask this question: can scaling up the amount of search we do by investing more computational resources help? So one of my lab mates, Chensi, took this sort of worked on this problem. And basically what he did was he took these protein diffusion models like Chroma and said, okay, what if we start just diffusing and generating a bunch of different protein backbones, scoring them with AlphaFold based on this sort of AlphaFold ranking methodology, and then using that to sort of evolve new structures that AlphaFold likes more and more and more, and seeing if we can use that to sort of converge on correct native structure predictions where AlphaFold fails in the single sequence regime. And what we found is that this works sometimes. So here's an example where you start with some sort of structural template, you perform these rounds of sort of iterative denoising, keeping the confident regions and sort of renoising the unconfident regions. So we found that, yeah, in some cases you can do this and you get structures that are more confident and more correct. But in other cases, you find these adversarial examples where the confidence goes up, but actually the structure that you converge on doesn't really look like the native structure. So this purple prediction doesn't even really look very much like a protein. It's not very compact, and it has all of these exposed surfaces. So that's a little puzzling. And what we think is going on here is that AlphaFold has been trained on a lot of weird proteins like single chains and cropped proteins. And we suspect that the model can sometimes implicitly infer the presence of sort of like a cropped out region or a binding partner. We didn't really encounter this when we were doing in the initial sort of AlphaFold ranking paper, because we were mostly using templates that have been generated by other structure prediction methods. So they were already like fairly well compacted. But you can see this in, for example, these examples where you have AlphaFold predict sort of like an isoform that has a lot of exposed hydrophobic residues. These are like isoforms that are like subsequences of myoglobin. And this, I think, is with an MSA, but AlphaFold will, in this case, happily predict this structure that has these exposed hydrophobics, which probably isn't that accurate for that real isoform that exists. So other issues with using AlphaFold 2 is this energy function. So AlphaFold 2 based scoring is quite slow, and filtering based on AlphaFold confidence metrics can actually be the slowest part of many modern protein design pipelines. And also, when we talk about AlphaFold having learned an energy or potential function, we're being pretty loose with this. This is not an energy in any mathematically rigorous sense. So we can't use this for like forces, dynamics. If we want to sort of simulate the process of folding, that's not something that we can do using some sort of energy function derived from AlphaFold. So basically, coming from this work, I was thinking about, how can we use machine learning to better model protein energy landscapes in a more sort of rigorous and in a more unified theoretical and on a sort of stronger theoretical foundation. And so I want to briefly talk about protein energy landscapes. So we have this idea that protein energy landscapes are dictated by the Boltzmann distribution, where you have some energy function that takes in both a structure and a sequence, and basically it's going to be Boltzmann distributed where structures at higher energy are going to be less likely. This in this case suggests a potential energy, but you can also integrate out various degrees of freedom, and this sort of becomes a free energy also. And so energy landscapes and energy functions are a really great way to describe protein conformational landscapes, because if you have a good model of sort of the energy landscape of a protein, you can do lots and lots of different things. So for instance, we talked about protein structure prediction. Protein structure prediction in this context just corresponds to finding a minimum energy structure subject to the energy function for a given sequence. We can also model conformational ensembles. So if you can sample from that Boltzmann distribution, then you can understand sort of the multitude of states that the protein can adopt. Sampling is its own challenge, but we can do that by running dynamics. So this is basically, if you look at the gradients of an energy function, those correspond to forces, or if it's a coarse-grained energy function, a potential of mean force that lets you sort of simulate the dynamics of that protein and sort of make movies of how it would behave. You can also use energy functions for doing things like stability prediction and binding affinity prediction. So to a very, very hand-wavy approximation, a folding free energy looks something like the difference in energy between a folded state and an unfolded state. This little note says that the real statistical physicists will cringe at this, but I'm hoping I can get away with it in this audience. And I'm primarily going to be using these heuristics when I talk about sort of differences in folding free energy, so differences upon mutation. And we can also do similar things for binding affinities. So we can look at differences between the energies of bound and unbound complexes and think about how that correlates with the affinity of sort of a ligand and target protein. So if you understand the energy landscape of a protein, you can really compute a lot of extremely useful quantities that we're really interested in modeling and predicting. So it's not a surprise that people have been trying to construct good models of protein energy landscapes for a long time. So in MD, basically what you do is you construct sort of a model called a force field based on the atomistic coordinates of protein, a lot of water generally in all atom MD, and also any other cofactors that are floating around. And you generally do so using a set of very cheap physics-motivated functional forms. So you have things like harmonic potentials for bond lengths and angles. You have specialized potentials for dihedrals, and then you have things like van der Waals potentials and Coulomb terms for electrostatics and lots of other stuff. You can fit these force fields to lots of different things. So you can fit them to QM data, experimental energies of folding, binding, and solvation. There's tons and tons of different fitting methods, and these force field parameters have kind of been accumulated over decades. There are lots of drawbacks, though, as anyone who's tried to use all of them MD will know. So force fields are a vast oversimplification. They generally mostly look at two-body terms. And you also need a very small step size to keep these simulations stable, because you're simulating things that occur on very, very fast timescales, like, for instance, like the vibration of a hydrogen atom. And so that means that simulations take forever, even though evaluating one step of a force field is extremely fast, extremely cheap. And you generally also need to model all the atoms in a system, which makes scaling to large systems difficult. So in the protein world, there's a very rich tradition of actually just using data on protein structures to parameterize energy functions. And there's a very long list of these sort of so-called statistical potentials that have used basically data on protein structures in the PDB to inform the design of potential functions. But one that people in this audience are probably very familiar with is the like Rosetta energy function, which has some MD-like terms. So it has all of these like harmonic terms for like bond angles and bond lengths, and well, actually it doesn't use those, but for like hydrogen bonds and things like that. But it has like these physics-derived implicit solivation energies and things like that. But it also has these statistical potential terms that say things like, okay, how do we parameterize the energies of sidechain rotamers? Well, we look at the distribution of rotamers in the PDB, and we use that to parameterize the distribution of sidechain rotamers. Same thing for backbone torsions. And also even I think the latest Rosetta energy function, the hydrogen bonding potential, is also based on a statistical potential. And so this has been really successful both in Rosetta and, like I said, the plethora of other statistical potentials that have used this sort of idea. And it's been successfully applied to structure prediction, sequence design, binder design, all of this stuff. These types of energy functions are generally not used with like the classical MD style simulation and are sampled with Monte Carlo. But there are still some drawbacks. So sampling can be slow. The functional form is still somewhat restricted in its expressivity in that you're still primarily looking at two-body things. And also, as we know for the past few years, these types of statistical potential methods have been outperformed in various tasks by more complicated ML models like AlphaFold 2, ProteinEBM, you name it, there's tons of them. So I guess that sort of drives this question of, well, can we marry sort of the power of data-driven potential functions for proteins with sort of the expressive magic of modern machine learning? Someone who actually recently tried to do that was like this no-name researcher guy over here. In his PhD thesis, he learned this. He created this energy function from the PDB that sort of learns pairwise radial distribution functions for all amino acid pairs, along with an ML-based rotamer packer. And this is fit via contrastive divergence, which is sort of this classical ML technique for fitting energy functions to data. And this is pretty cool, and it works pretty well. You can use it to do folding simulations of unfolded chains to their native states. And yeah, so this is John Jumper's PhD thesis. If you have some time, give it a read. It's pretty cool. So basically, building off of this long, rich history of building energy-based models of proteins, I thought, okay, well, how can we sort of marry, that sort of successful paradigm with some of the really powerful tools for protein ML that have come out in the past couple of years. And so first, a brief overview of energy-based models. So energy-based models are just a class of machine learning models that try to learn an energy function. And what we mean by energy function is just that they're trying to approximate the log of the probability density function of a certain distribution. In this case, we're going to be talking about the distribution of protein conformations X conditional on protein sequences S. And also in this case, we're going to be looking at a coarse grain view of this. So we're going to be looking only at the C alpha coordinates, although that can be generalized. And so training an energy-based model on sequence structure pairs could give us the energy that we want, something that sort of resembles the free energy landscape of a protein. Although that's subject to the significant caveat that we're actually going to be approximating. We're not actually training on equilibrium samples here. We're training on stuff from the PDB. But like I said, this sort of assumption is kind of inherent in all of the work that uses data-driven potentials for proteins. And we can also potentially mitigate some of that bias by fine-tuning on MD trajectories. And so, okay, how do you train an energy-based model? Well, we already mentioned contrastive divergence, which is what Upside did. A more modern way to train energy-based models is this denoising score matching idea. So the denoising score matching comes from the idea that comes from the fact that one way to make two distributions equal is to make their scores or their gradients equal everywhere. And so basically what we want to do in this paradigm is we want to take our sort of neural network energy function E and its gradient with respect to its inputs and match that to the gradient of this probability density of the data. But we don't actually know what the probability density of the data is. So there's this neat trick that you can use to make this tractable where you take the data distribution and you add like a little bit of noise, in this case Gaussian noise, to it. And then you have this relationship called Tweedy's formula, which basically says that actually the gradient of the noise distribution is now related to sort of this denoising estimator, or the expected value of the denoised data conditioned on the noisy data point. So basically, what this is saying is that the score of the noisy distribution, this gradient, should pull you from a noisy data point back to sort of the expected unnoisy data point. And this relationship is called Tweedy's formula. You can use that to create a training loss, which basically says, okay, we want to enforce this property, except we want to sub in here our learned energy function. And we want to make it equal to basically the denoised data point. And so that might seem complicated, but really what I'm hiding behind all of this math stuff is that this is just a diffusion model. So this is exactly the training paradigm of diffusion models, except in this case, what we're doing is we're taking the denoiser and we're parameterizing it as the gradient of an energy function. So don't worry if that was confusing, because I think this next explanation will be a lot less confusing. So in a traditional denoising diffusion model, like RF diffusion, what you do is you have a noisy distribution. We might call that t equals 1. And then basically we have a model that is going to iteratively denoise that from t equals 1 to 0 until we arrive at something that looks like it came from our data distribution. So yeah, so if this is something like RF diffusion, we have this noisy input, we have this time parameter, and then we're trying to predict the clean input. Instead, what we're doing here is, instead of predicting the clean input, we're just predicting some number, right? And what do we do with that number? Well, we're going to differentiate it with respect to the input to get this gradient. And then we actually want this gradient to be what takes the noisy input back to the clean input. So that value is our energy, and this gradient is the force, right? And so what all that fancy math on this previous slide said is that actually, if we do this procedure at low levels of t, this E function should actually match up to a constant, the data density of the training data. Here's another way of visualizing that. So basically, we have these noisy structures, and we're going to noise them up. And basically, we're trying to train an energy function that's going to pull those noisy structures back to the unnoisy structures. That's really all we're doing here. So it's a pretty intuitive thing to do, but it's very powerful in that at these low time levels, in theory, this should converge to something that approximates the data density. And so how this is different from a normal diffusion model is that at inference time, we can actually take that energy function and do lots of things with it. So we can input a protein structure and see how the energy changes when we manipulate it somehow. Yeah, and so basically the way that we train this is we train it on a bunch of domains from CAF, some AFDB structures, some two-chain complexes. There's two chains. And we fine-tune it with some MD frames that were published by the BioEMU paper from Microsoft. And basically, this is just to help sort of mitigate that bias that I mentioned earlier, where we're sort of, we're not actually training on equilibrium samples, we're training on things from the PDB or the FDB. Yeah.
Right, but the pre-trained stuff?
Yeah. Yeah.
You have to go back for a given X then in the pre-training part, and then you
Yes, correct. Yeah. Okay, so I mentioned that, if we do this procedure and we fit a model that's going to approximate the log of the data density, we should in theory have something that might meaningfully resemble a free energy function for a protein. But this actually is subject to a whole bunch of caveats. There's no actual guarantee that that's true. There's no theorems in deep learning, right? So ultimately, it's an empirical question whether doing this actually gives us an energy function that's at all useful. So what are some things that we can do with an energy function trained this way? Well, one sort of fundamental capability and kind of a sanity check that this model is doing anything is, can it do that scoring thing that we mentioned earlier from AlphaFold? So if we have a bunch of protein structures of different distances to the native structure, are the low energy ones closer to the native structure? And we see that this works really well. So this is an example of a protein where we have a bunch of decoys that are at different TM scores to the native structure, and the ones that are more accurate are given lower energies in our energy function. And if we actually look at the correlations here, or I should back up. This model takes as input a protein structure, and it also takes in this time parameter. And so basically, in a diffusion model, that's telling you how much noise you're trying to remove. And in this case, that's still sort of what it's doing, but it's basically you're learning a bunch of different energy landscapes, which have a bunch of different amounts of smoothing. In this picture, basically that's illustrated by the fact that these low noise energy landscapes are going to be much more rugged. And then as you go up towards t=1, they become much more smooth and actually converge to a Gaussian density. So we treat this as sort of like an empirical hyperparameter, like what T index energy landscape actually performs the best. And we find that actually it sort of peaks around these low but non-zero values. And I think that this actually makes a lot of sense because if you have a very low value of T, what the model is basically learning to do is just sort of fix local features. And so it might not care much about the global features of the protein structure, like, like how much hydrophobic surface area have you varied or something like that. On the other hand, when you go to t=1, you're basically converging to a Gaussian density. So you're basically just going to be ranking structures based on their radius of gyration. So the sweet spot somewhere in the middle is like t=0.05 for this model. Yeah.
So I guess I'm wondering if you looked at different times.
Yeah.
And then took a weighted.
Yeah.
Some of them.
Yeah.
It's better, or are these all
Yeah.
correlated? Or?
I've tried that, and you can improve it by doing that. I just went with a single one just because it was a simpler thing to do. But yeah. And so actually another thing that we found was that, so we trained this model and we found that we could do a really good job ranking these structures. This solid black line here is the Rosetta energy function. So with the right time level, we're beating the Rosetta energy function at ranking quality. And then actually I thought, well, okay, if this is the best, what if we sort of concentrate the model capacity just on these lower times? So basically what we did was that we then trained a model on just these lower times to create this so-called expert model that does even better. And so, yeah, so we can look at scatter plots of how well it ranks these targets versus the Rosetta energy function, and it's better on most of them. This is including, so these easy targets are split from the training set with a sequence similarity cutoff, and the hard ones are split with a fold cutoff. So these are folds that the model has never seen. And so even on the unseen folds, it does a really good job with the ranking. And that's the same if you look at it in terms of correlations, or if you look at something that's maybe a bit more practical, which is like the top one. So like how accurate is the top lowest energy thing that you've picked up? So this, I think, is a good sanity check that energy function trained this way does a good job discriminating good protein structures from bad protein structures. And so another thing that we can do then is look at mutation effects. So how does the energy function change when we substitute a residue? So the way we're doing this, the way we're going to approximate this here is we're going to look at the change in energy upon mutation for the folded structure and also for a model of the unfolded structure. And basically, this is because this energy is unnormalized. So you can have changes in energy just based on sequence that are kind of arbitrary. You need to kind of subtract out the background by having some approximation of what that mutation would do in the unfolded baseline. Rosetta, for those of you who know about Rosetta has this as like the reference energy, for instance. So this is coming up with the reference energy. And then so to look at the change in stability upon mutation, basically we take this difference for the wild type between the folded and the unfolded. We take this difference for the mutant between the folded and the unfolded, and we look at the difference. And so this does a really good job at predicting experimental delta Gs. This is from Gabe Rockland's data, which uses the cDNA display technique. And so this is an example, and this actually is a comparison with ESM3, which is one of the sort of state-of-the-art protein language models for this task. And yeah, we're generally better. Well, we're better than ESM3 on this, which is interesting because ESM3 is much larger, and it's also heavily utilizing sort of evolutionary information based on sort of residue conservation within those proteins. And actually this gap, and so actually if you look at the protein gym stability benchmark, this model is number one, but I mean, more than just having some marginal gain on a leaderboard, I think this is also pretty cool because this is a pretty big departure from the rest of the models on here, which are all protein language models. And this is like an energy-based diffusion model, which I think is it's interesting. It's cool that, a sort of a new modeling paradigm does so well on this task. And the gap actually gets even bigger when we look at proteins that don't have a lot of evolutionary information. So as I mentioned, the protein language models, they can really tap into residue conservation and residue interaction that you might find in an MSA to reason about fitness effect prediction. So So if we break down these sort of protein gym or like mega scale targets by how many how many MM-seq hits they have, the ones that have very few hits, so these are likely de novo proteins, ProteinEBM is way better than these two baselines, ESN3 and ESM-IF1. And for ProteinEBM, actually, the performance on this stability prediction task doesn't even really depend on the amount of evolutionary information. Whereas when you look at the protein language models, it drops off a lot for the proteins that have few homologs. So that's pretty cool. And I think that sort of confirms that, an energy function trained this way can be useful at tasks that it wasn't explicitly trained for. Again, this is definitely never trained on anything like this, like trying to sort of predict what the fitness effects of mutations. Yeah.
I keep asking much, but
Yeah.
So does your model consider side chains at all, or is it just like a backbone frame?
Yeah, so it's just backbone representation, but it can implicitly sort of model side chains. Yes. And there is actually an auxiliary loss for predicting side chains. So it knows about side chains, but the side chains are not like the side chain rotamers are not inputs to the model. Yeah. Yeah.
What about predicting of absolute folding energy?
Yeah. Sure.
You that look a bunch of unfolded.
Yeah.
How much does that matter?
It doesn't matter a ton, but it does help. Yeah. And so also you mentioned absolute folding energy. So we're not doing absolute folding energies here. These are relative folding energies. So delta G's. This approximation where you just sort of naively subtract a bunch of energies, that wouldn't really work for computing an actual delta G. Yeah. And so, okay, so we can also, another thing that you might want to do with sort of a learned potential function is explore the energy surface, right? So with this model, we have a couple of different ways to run dynamical simulations. We can treat it as a diffusion model where we just start from noise and do this sort of reverse time diffusion and generate samples from our data distribution. Or we can sort of we can run dynamics at a given step. So we can literally just follow the gradient as you would in sort of Langevin dynamics. You take gradient steps, you add some noise, you take gradient steps. And you can also combine the two. So we can also bounce around with Langevin dynamics on a given energy level and then bounce down and do some more steps. So there's a lot of options when running dynamics here. This is running dynamics through this Langevin annealing protocol where we basically take a bunch of steps at each time level and then we go down a noise level until we get to t equals 0. And what we can do here, which is cool, is that we can generate a bunch of samples and then we can rank them all by energy. So for those of you who've worked with like Rosetta before, these are similar to those sort of folding funnels that you see from the Rosetta, from the like Rosetta ab initio protocol. And so these are these fast folders that have been run in all atom MD. And what we see is that when we run these, we get good agreement with the experimental structures. We get some exploration away from the native structure, although not as much as all atom MD, which I guess is kind of a consequence of the fact that we're mostly training on native structures. And also these simulations will run at higher temperature. But and also sometimes we sample a lot of a lot of structures that actually are not correct. So a lot of structures away from the native structure. But using the energy, we can pick out the good structures. Right. These are also all held out from the training data. And then we can also do direct folding simulations where instead of starting from noise, we just use sort of this low energy level and just directly run Langevin dynamics starting from an unfolded chain. And we can make movies like this where we start from an unfolded chain and then we can watch the protein fold up, which is pretty cool. This, you could do with all atom MD, but this is like very fast. This takes like GPU minutes. And then we see that, this is not biased in any way towards folding to the native structure. This is just letting the energy do what it's doing. And when we do this, we can make a movie of the protein folding from an unfolded state. Here's another movie because these are fun. Oh, wait, of a different protein. This is a fast folder NTL9, and you can see that as it folds up, the RMSD goes down and also the energy goes down until you reach a point where you have an accurate structure that has a low energy. This animation was made with Pi2D-Mol, Sergey's fancy new 2D protein visualization suite. If you want 2D proteins, you should check it out down here. So that's pretty cool. But are these pathways accurate? Well, how can we compare these with experiments? One way to do this is with something called phi-value analysis. So phi values are an experimental measure of the order in which residues fold. And basically, the experiment is you take a protein, you pick a residue, you mutate that residue to alanine, and you measure the folding and unfolded rates. And then you compute this value phi. That's based on the unfolding and folding rates of the mutant and wild type. And so basically what you're doing here is if you visualize like the folding landscape of this protein, you have the unfolded state, the folded state, and you have a transition state, and then the folding and unfolding rates are going to be driven by these barrier heights here. When you make a mutation, right, say this mutation raises the energy of the transition state and it raises the energy of the folding state by about equal amounts, right? Well, then this mutation is going to affect the folding rate a lot, but it's not going to affect the unfolding rate very much because it just kind of got translated up altogether. And so when you plug that into this formula, you get a value of 1. And what this conceptually means is that we think that the residue is ordered at the transition state, right? Because basically the amount at which the transition state was destabilized was the same as the amount which mutation destabilized the folded state. So this indicates that the residue is maybe in a native-like conformational space at the transition state. Conversely, if you have a mutation that doesn't affect the transition state very much, but does destabilize the folded state, then if you plug these values in, you'll get a phi value of 0. So this is taken to mean that the residue is not very ordered at the transition state because mutating it destabilizes the folded state, but it doesn't destabilize the transition state as much. So yeah, generally, phi close to 1 indicates folding early, close to 0 means folding late. You can approximate phi from simulations by computing the fraction of residues the fraction of its native contacts that a given residue forms along like a transition path. The interpretation of these experimental phi values is noisy and subject to a lot of caveats, but generally it gives us a decent sense of how well of the order in which residues fold. So then we can actually take our simulations and compare to experiments. So this is ubiquitin. In this case, the phi value suggests the N-terminus folds first. That's also what ProteinEBM thinks. Although these C-terminal phi values, I think the ProteinEBM ones are a little large. But generally, the trend of the N-terminus folding first is correct. This is a small WW domain. We get the same general shape as the phi values. And also, we can compute phi values from all atom simulations, and they also agree fairly well. This is protein G, that one that I just showed in the movie. In this case, the C-terminus folds first, and we see that also in both the experimental phi values and the ProteinEBM data. And so I should mention now that one really big test of whether this works is actually whether it can distinguish differences in folding between two proteins that have the same topology. Because a lot of times, if topology is constraining the folding pathway, you can actually predict the folding pathway largely just based on the geometry and not really considering the explicit side chain interactions, right? But so in these hard cases, like protein G, people have actually, like the Baker lab back in a long time, like 20 years ago, engineered this mutant version of it that actually was supposed to have a reversed folding pathway. So this version folds through the C-terminus, then they engineered this other version that folds through the N-terminus. And basically, when we run those two with ProteinEBM, we also see the reverse folding order as well, which is consistent with the experimental phi values. And there's also a natural structural homologue of these proteins, protein L, that also folds to the N-terminus. This one, ProteinEBM doesn't get quite as right. It still predicts the C-terminus to fold too early, although it does capture the correct shift away from the structural homologue protein G. So the G versus L is very N-terminally shifted. That is captured by the ProteinEBM simulations, although not quite enough compared with the experiments. So that's our study of protein folding, which is something that we're currently expanding upon as we revise this work. But yeah, I think it's really cool that this is a computationally efficient method that can generate these folding trajectories, and we can compare them with experiments. And one thing that I'm really excited about for the future is actually using this to try and test and predict hypotheses about how proteins fold. And I think that
For any metamorphic proteins yet?
Yeah, actually, I have a whole section on metamorphic proteins if I have time to get there. Yeah. So a last thing that I'll talk about is structure prediction. So one thing that one way that I motivated this whole thing was trying to use this for structure prediction. So if we have a good energy function, we can sample from it and we can try to find good structure predictions for proteins, ideally from single sequence, right? And so this is how you would do structure prediction with ProteinEBM. You basically run lots of samples, and then you look at the lowest energy ones, and you say those are our structure predictions, right? And we want to see how good are those. We also developed this really accurate AlphaFold ranking system back in those early slides. And so another thing we can do is try to combine the two. We can take the good scores from ProteinEBM and apply them to the AlphaFold scoring and re-rank them. The reason you might do this is because I don't know, it's like sort of ensembling two methods, and also AlphaFold is very slow, but if you only use the top scores from ProteinEBM, then it doesn't really matter that it's slow because you're only running a couple of them. But this basically these are a couple of structure prediction workflows that we tried. And so here's how this performs. So basically, if we look at things that are, so basically we use ProteinEBM sampling and we compare it against compute match baselines of AlphaFold 2 and AlphaFold 3. So we ran tons of samples with AlphaFold 2 and AlphaFold 3 to basically make sure that we were using as much, or actually in this case, a significant amount more compute for the AlphaFold baselines than ProteinEBM. And also these targets have been held out from ProteinEBM, but they haven't been held out from AlphaFold 2 and AlphaFold 3. So this isn't exactly a fair comparison. But we see that for these sequence split targets, ProteinEBM outperforms both AlphaFold 2 and AlphaFold 3, although not by an enormous margin. And then if we combine it with AF2 rank, it gets even better. And then for these but for the ones that are held out, like the whole fold is held out from training, we still see that it's difficult to sample the structures there. So if we look at these scatter plots, yeah, we're definitely better than AlphaFold on the sequence splits. But when we actually held out the structure from training, it can still be difficult to sample these targets. So we haven't quite solved everything yet, but we're working on it. Here's an example of one where we sort of failed to sample the correct structure. And oftentimes we see, as kind of we expect, this isn't really a ranking failure. If we initialize it with the native structure, we get that scored as the lowest energy. But actually, when we just sample with ProteinEBM, we didn't quite reach there, and we sampled these conformations that sort of have this helix in the wrong orientation. But yeah, so this suggests that if we go way back to the first slides, ProteinEBM did a really good job at the ranking task, even for these proteins where the fold was entirely held out. So this suggests that we can rank unknown folds quite well, but still sampling them remains a challenge. Yeah, and then so I guess with the remaining time, I'd like to talk about using ProteinEBM for design. So mostly this talk was about modeling, but this is Boston Protein Design and, well, protein. I guess the design comes first, but I guess we're flipping that. So energy functions have been used for design for a long time, and this one is no exception. We can also use it for design. So Gabe, who rotated in our lab this spring and who now is joining, he worked on using ProteinEBM for sequence design. And basically, the protocol here is extremely simple. You pass ProteinEBM a structure, and then you just try to minimize the energy with respect to the sequence, and you see what comes out. And so this actually works. We can get decent native sequence recoveries, and in silico, if we refold the sequences, they fold and AlphaFold to the right thing. If we run sampling with ProteinEBM, they also fold to the right thing. And then we can also extend this work to also basically perform gradient descent with respect to sequence and structure at the same time. So ProteinEBM's inputs are, the time parameter, a structure, and a sequence, and basically we can just compute the energy and backprop to all of these things and slowly move the time from 1 to 0. And we do that, it looks something like this. So we start with spaghetti structure, and then we run gradient descent on both the energy and the sequence space. And gradually sort of the sequence comes into focus, turning into sort of like a crisp sequence. The energy goes down and we get a protein structure out of it. So this is sort of and then if we take the sequence and give it to AlphaFold 2, then we get the right fold back. So this is sort of an example of sequence structure co-design, where we don't need to use like protein MPNN or anything like that. But we also don't have to train like a specific sequence generator. We sort of get it for free by just optimizing this energy. So what if we want to go even further than that? One nice thing about this energy parameterization is that we can potentially try to make the energies of different states equal. So this is sort of like what you would get with a metamorphic protein. And so this sort of convoluted diagram, which is one of things from papers from our lab often have diagrams like this. If you've seen a talk on Minecraft or something, or AF Cycler or something, there's arrows going everywhere. But this really isn't that complicated. What we're basically doing is we're repeating the same design workflow, except we're doing it twice with a shared sort of sequence input. So we have a single sequence that we're trying to reduce the energy of with respect to both the sequence and two distinct sets of coordinates. And then we don't want, but if we just did that naively, we would get the same sequence, we just get the same structure twice. So what we're also doing is we have a secondary structure prediction head here, and we're trying to just push the secondary structures apart while we do the same thing. And so ideally what this gives us is a sequence that can match two secondary structures at the same time. Yeah.
But I'm saying that's the sequence structure pair with the least possible amount of energy, but all possible sequences. Or is it that straight somehow?
Do you mean just for the general design task?
Yeah.
Like this one? Yeah. So the least amount of energy, I guess in theory, yes, but I don't think we're actually going to get there. Like this is a very large search space, right? So if you initialize this a bunch of different times, you'll get a bunch of different things, right? But I think what generally you're looking for when descending the space is a compatible sequence and structure, starting from some random initialization, right? So it almost certainly is not the global optimum of all sequence and structure pairs, but it is a local optimum, I think. I mean, I think that you probably pretty quickly get trapped, right? Because in the structure design, right, there's so many different structures that you can find. The denoising protocol will sort of quickly get you in some local funnel, right? And then you're going to be sort of co-designing sequence and structure in that local area. Yeah, but yes.
Sort of, since you're on the slide, when you're updating the sequence,
Yeah.
is it in some kind of continuous sequence space?
Yeah. This is just like
at some point you have to make it a hard sequence.
Yeah. This is just like like Minecraft style type thing.
Okay.
Yeah.
So how do you make it a hard sequence?
You just take the argmax at the end. Yeah. Yeah, that's how we give it to AlphaFold. So you just take the argmax at the end. Yeah. So when we do this protocol, this actually works in silico. So we have this protocol where we can hallucinate this. So we can hallucinate these two design models, which share a sequence. And then when we give the sequence to AlphaFold, 3 of the AlphaFold models, like this one, predict this fold, and 2 of the AlphaFold models predict this fold. So we thought that was kind of interesting, and this might be a first step towards designing proteins that can sort of adopt multiple conformations at equilibrium. Because if we're using AlphaFold as our sort of in silico filter, well, the AlphaFold models are kind of split, like half of them like this one and half of them like this one. So we're excited about this as like a potential way to design interesting multi-state proteins. We've done a little bit of characterization. This is probably the worst experimental data that you'll ever see in a presentation at this club, but I'm a computationalist and I did some pipetting, so I'm kind of proud. So here's my CV curves. This protein was predicted to sort of have like these two states. We see some difference between the multi-state design and one that was sort of mutated to be locked into another one. This third curve is kind of too noisy to really tell what's going on, but we're working on characterizing some of these. So stay tuned for more results on that. Yeah.
Wait, so are those different sequences then?
Those are different sequences where basically we designed this one sort of chameleonic sequence, and then as controls, I sort of made mutations that were predicted to lock it into one state or the other. Yeah. Okay, I said if we had time, I would talk a little bit about model interpretability. I guess I have a couple minutes, so I will briefly talk about it. So classical force fields can be decomposed into these physically interpretable terms. So, if we have a classical energy, we generally look at it as the sum of like bonded terms, van der Waals, electrostatics, solvation, H bonds, etc. That's really nice because it lets us sort of play with, it lets us adjust the force field, and it also lets us know what's driving a given, like prediction. So is there a way that we can sort of try to do this with a learned entity function? And this is work that I've been wanting to do a long time. I was kind of spurred into doing it recently by reviewer number one. My thanks to them. So here's a quick thing we can try doing. What if we just like sort of make two like helices with residues and try to like incrementally bring them together? What does the model do? Well, we can look at the energies, and we can see that for different, basically we get something that kind of looks like, sort of an attractive, repulsive Leonard-Jones term, with a couple of differences. So we see these sort of long tails where we would expect an LJ to just sort of flatten out. The long tails are the result of the fact that in regions where the data are sparse, we have this sort of Gaussian prior. So it should sort of just at very long tails converge to just sort of a quadratic potential that kind of pulls stuff together. But that should be dominated by other terms once we get closer in. And yeah, so we have this repulsive term too, so that once we get close enough, the energy spikes as you bring it closer together. And those preferred distances make sense. So more charged and larger amino acids, generally like more space from themselves when they're packing together. And this is also pretty well correlated with, empirical distance distributions between amino acids that you find in the PDB, although these are kind of systematically lower. So it seems like the model it's encoding repulsive van der Waals and is sensitive to clashes, but maybe not quite as much as you would actually expect. And I think this kind of makes sense because the model has been trained on sort of a loosened, it's sort of modeling a noisy energy landscape that's kind of been smoothed over. So these really sharp things like, like the like power of 12 or power of 6 term and a Leonard-Jones potential, those might be sort of softened and made kind of less sensitive. Another thing we can try to do is to probe sort of how the model encodes like physics is to look at mutations and specifically to look at epistasis. So here we're looking at making multiple mutations and then looking at where sort of the sum of making 2 mutations is different from making those 2 mutations individually. So this is like a map of where we see epistasis on the protein when we make pairs of mutations and look at the combined energy minus the individual contributions. And we see that this basically maps perfectly to the contact map of the protein, which makes sense. We expect epistasis to exist at residues that are in contact. And if we actually look at the patterns of epistasis, so for instance, these are 2 residues that are engaged in a buried hydrogen bond. One, we can see very strong epistasis for charged residues that you would expect for charge complementarity. So, it's very epistatically favorable to choose one negatively charged and one positively charged amino acid, whereas, like charges are very unfavorable. And we also see positive sign epistasis for mutating to a hydrophobic residue. So basically, this buried H bond pair, if you mutate just one of them to a hydrophobic, that's destabilizing. Those are all. But then if you've already mutated one, now it's stabilizing to mutate the other one to a hydrophobic, which makes sense, right? If you get rid of one H bonding partner, then it's favorable to mutate the other one to a hydrophobic residue. But a priori, it's not favorable to mutate either of them to a hydrophobic residue. So these are just some early investigations of sort of being able to interpret how the model reacts to different inputs. But generally, the punchline so far is that the model seems to understand, the general contours of like hydrogen bonding and charge complementarity and van der Waals, although it seems less sensitive to sort of these like sort of atomic, like for instance, things like sidechain rotamers and repulsive van der Waals the model seems a bit undertuned to, which I think makes sense and motivates maybe going towards models that can also represent all of the atoms explicitly. Cool. And I think that's about, oh yeah, that's the synapses map. Oh yeah, and then here's with if you have 2 hydrophobic contacts, we see some epistasis for introducing multiple charge complementarity or hydrogen bond donor acceptor pairs. And we don't see really any like sign epistasis except for one for introducing a disulfide, which also makes sense. Yeah.
Yeah, there's what you might see from an AlphaFold in terms of detecting epistasis.
Oh, that's a good question. No, I don't at the moment, but I'll definitely look into that. I mean, generally, AlphaFold confidences are not super well correlated with the destabilization of proteins, just because it's so attuned to sort of the global fold that like disrupting one residue, like it just kind of will adjust the local geometry and not the confidence metrics. You won't necessarily see a huge change. But yeah, that's definitely worth looking into. Yeah, and so with that, I think we can talk about conclusions and future directions. So so energy-based models of proteins have a long, successful history. Basically, the goal of this project was to combine the flexibility of EBMs with the sort of expressive power of modern machine learning. And I think we shown that can be a useful paradigm that leads to like a pretty versatile model. And areas for exciting future applications are like trying to study protein folding dynamics. One thing that I'm interested in is looking at the difference in protein folding dynamics between de novo and natural proteins, which I would love to study at scale. Gabe is interested in designing transition paths. As I mentioned, expanding modeling to all atom resolution would be cool. And yeah, I'd like to say thanks to my lab. Thanks to everyone who's helped with this project and people outside of my lab who've helped with this project and all of our funding sources. Here's some pictures of us having fun. I'm really grateful to Sergey and the entire lab for being a great place to be a student and do a PhD. So thank you all. Yeah. No.
Super cool. So I have one part preamble, one part question.
Okay. Yeah.
So for Gabe Rockland's data.
Yeah.
And you're fitting the delta G's.
Yeah.
Obviously you do a really good job.
Yeah.
Good correlation, but
yeah.
you miss some. And so the preamble is I'm assuming that you're using your fine-tuned model that's doing little time steps sort of from a starting structure. And
yeah.
You're assuming, I think, that Gabe's structures that he designed sequences for are the starting structures that are appropriate, I think. And so my question is this, that if you don't assume that, like you take some of these points that you didn't get right.
Yeah, sure.
And if you do the folding simulation, fold it and try to find free energy of a folded state, maybe these don't fold to that structure.
Yeah, right? That's possible, for sure.
I wonder if you would get a better correlation if you did that for some of these points that
totally. I mean that is a good idea, and that's generally what you would do if you were trying to do this like with MD, right? You wouldn't just like evaluate an energy change, you would sample it, right? The great thing about doing it this way is that it's super fast, like it takes like fractions of a fraction of a second, as opposed to actually like sampling the structure change takes longer, but we can definitely try that. Yeah, for sure.
But you have it. The second question I had was, do you have an idea of the error bars in this? Like if you do dropouts or something and then compute the energy like 100 times.
Yeah.
Do you get a spread?
Yes. And actually there's an even simpler way to do it. This model is based on the diffusion module from AlphaFold 3, so it's non-equivariant. So you can also just like rotate the thing, and that's sort of a randomization method. Basically, if you look at like these, and it's not that severe. So basically, if you look at what am I looking for? I'm looking for, sorry. Oh yeah, this slide. If you look at these rankings, like all of these decoys are like independently rotated and translated, right, with like a random data augmentation. So it doesn't really affect things that much. But there is some spread. I don't have a number off the top of my head, but yeah. Yeah. Yeah, sure, yeah. Yeah, so yes, we have done that. I have models that have been trained on multimers that we've used for predicting delta G's of binding. And it works pretty well. We're still trying to make it so it works about as well as, like, for instance, you can use like ProteinEBM to do this. It works about that well. We're still like, it's still an area of trying to improve that we're trying to improve on. And yeah, I think that there's probably a lot of ways to explore that. And yeah, so we've been working with various people to explore how well it works for binding. And yeah, it does seem to work, but I think there's still more work to be done. Yeah. Yeah.
I missed a fundamental point.
Okay.
What's the distinction between the gradient of the logarithm of the probability distribution, the potential mean force.
Yeah.
And which is what is used in the diffusion models to
Sure.
propagate.
Yeah.
And your energy function. And then, so I missed that distinction.
Yeah.
But then, so then that informs my question, which is, if you play a movie of the diffusion
Yeah.
process, like we do in chroma,
Yeah.
and I play your movie,
Yeah.
are they different?
No, they're not different at all. So that
So
part
then-
is the exact same. So what we did was we parameterized the well, first of all, chroma is sequence unconditional. So this is informed by the sequence, right? So this is taking in a sequence and trying to
We don't
model.
need bone protection.
Yeah, yeah,
Diffusion.
yeah, yeah, yeah. Yeah. So in a diffusion, in a normal diffusion model, basically, you're just sort of directly predicting this denoise structure, right? Instead, what we're doing is we're parameterizing that as the gradient of this energy function, right? So chroma, for instance, doesn't have an energy function. It just predicts gradients, right? It just predicts them like straight away.
The gradient is there, and that's an equality.
Well,
Mean force. Gradient.
Sure. But you can't, if you wanted to actually access there is an implicit potential function,
That's what it's for.
but you can't access it to do anything with it, right? If you wanted to look at the implicit,
If I do it in 200 time steps and I stop between 99 and 100, then I directly access.
no, you don't, because you have the gradient, but you don't have what that gradient is the derivative of, right? I mean, you could try to integrate it somehow, but basically, if you wanted to use that to, for instance, say which chroma samples are lower energy than the others, you can't do that with chroma, right? Because it's just parameterized as the gradient is the direct output of the neural network. It's not parameterized as yeah.
But then, the dynamics, you say, would be the same.
The dynamics of the reverse diffusion process, yes. Although I think the way we've trained the model makes it better at doing things like this. That's mostly has to do with the training data. But yeah. Great. Yes. Yeah.
Oh, great. There's been some work published recently talking about mutational invariance in AlphaFold. Whenever you mutate a ton of the sequence and the structure doesn't really change.
Sure. Yeah.
Consider taking in a conditioned AlphaFold structure that's a wild type and then changing the sequence and seeing if your potentials can help it find the actual fold under those gross mutations.
Yeah. Sure. I mean, I think it's difficult to say that whether the mutations that you're going to do to a big, like if you're going to make a bunch of mutations to AlphaFold, like if you're going to take an AlphaFold input and mutate 50% of the residues to alanines, I don't really know if it probably doesn't necessarily have a new structure. It probably is mostly just destabilized and more unfolded. But yes, you definitely could do that. You could definitely run dynamics, run simulations with this model, see what the new lower energy structures are, for sure. Yeah.
Just a quick follow-up to that.
Yeah.
When you give it some sort of nonsensical sequence that the potentials don't really have a good answer for, does it fail gracefully? Is it able to identify that there really is no folded structure? That's
a good question. I don't actually have a great answer for that. I mean, we can definitely find unfolded structures. Like for instance, if you look at the samples of these guys, we do sample unfolded areas of the phase space, but that is a good idea of like testing, like IDRs or something like that. Because IDRs are very far from, we haven't trained on any IDRs, obviously, but it'd be interesting to see what happens. Yeah. I mean, I think it's quite possible that, like in other potential functions that are mostly trained on native structures, it would maybe not fold into something, but at least probably be over condensed. But that'd be very interesting to try. Yeah. Yes.
For structures that most diffusion models get wrong,
Yeah.
have you tried putting both the crystal structure and the prediction into the energy function and seeing if maybe the crystal structure is lower than that?
Yeah. So yes, and yeah, that does work. Like for instance, if we look at for instance, in this case, even though the model didn't actually sample this correct conformation and sample these, somewhat wrong conformations. If you do give the model the native structure, which is this, the energy is lower. So yes, I suspect that would work. Yes.
In this example, did you run the sequence through a diffusion model, like and see where it fell?
Not exactly, but I mean, I think that if you use a naive AlphaFold single sequence, it would be somewhere low. Yeah. Yes.
Great talk. How easily, say, framework be modified to accommodate small volume?
Oh, very easily. Yeah. I mean, you just kind of do it. But yeah, I mean, some yeah, I mean, I'm not exactly sure. I mean, in theory, yes. I think it might introduce some subtleties in terms of how well, yeah, I mean, I don't know. We should just do it and see. Yeah. Yes, Jody.
So why did you choose to have it diffused into a Gaussian? That's the Gaussian. There's other ways.
Oh yeah. I experimented with the chroma version. And it didn't really, I don't know, it seemed my initial results weren't great. And so I was just like, this is the simplest thing that everyone does. So I'll just start with that. Mathematically, it kind of doesn't really matter because once you think about sort of the time and the variance converging to zero, the process should converge either way. But yes, that is probably something to look into. And I have looked into it, but it's probably something worth revisiting. Um, yeah. No, that's definitely a good intuition, something that I've thought about a lot. Yes, I think that is a good idea. I mean, the Gaussian comes with the benefit that it's easier to train. If you do something too fancy, like if you're just trying to unfold it like with like a something that looks more like a real potential, then that becomes more difficult to train because like you need like a sort of simulation-based training. But yeah, the chroma-based process is not too hard. And like I said, I have tried it and it didn't really, but that was a long time ago, so I should probably try it again. Yes.
Okay, hydrogen bonds on the.
Yeah. Yeah.
Is that real thing? Are you sure about that?
Oh, well, in MD,
for
no.
5 years, people have been arguing that electrostatics and van der Waals should cover that.
Oh, yeah. In all MD, no, definitely not. Like Rosetta has a hydrogen bond potential, right? So that's I threw that in there. Yeah.
But then is there, I mean, I argued that you sure I was about.
Yeah.
Whether there should actually be a hydrogen bond.
Yeah.
Does that describe any of the complexity of the shapes that you were showing towards the diffusions?
On the first point of whether all AutoMD should have a hydrogen bond potential, I mean, it's hard because then you have to define bonds, right? And so generally in MD, you keep the bonds fixed. But I guess that's so what was the second question? I don't think I fully.
There are some shapes of your distributions, right? They were not harmonic.
Oh, yeah. Oh, these.
Leading to this,
Oh, well, I don't think you would expect those to be harmonic. So those are more similar to what you would expect from like a, like sort of Van der Waals type thing where you wouldn't expect a harmonic potential. But oh, whoa, okay. Yeah. So I think I was meaning what I was saying is this long range attractive thing sort of comes from the Gaussian prior, I think, which sort of just brings things at long range together, which isn't physically realistic, but is expected based on the mathematical formulation.
Yeah, is there any data that you have and that's informing these folding trajectories that you have, right? You have noise that you've been adding to these.
Yeah, sure.
It's like how expanded or unfolded does
so the noisy so the noising process does not create unfolded structures. The noising process is purely like a non-physical Gaussian corruption. The idea is that by learning to sort of correct that and match the score, you can still learn useful terms that can help you sort of guide a folding process from a more physical denatured state. And we also do train on some MD data that has unfolded states in it.
Be a direct conflict with cooperative folding, right? And suggest suggesting that folding is a locally driven process?
It would be a direct conflict with cooperative folding. Well, I mean, I guess folding is, I mean, you mean the Gaussian noising schedule would be a direct, because like the high noise states don't factor into the simulations of folding, right? That's all done at the low noise potential, right? So that's all done with this low time level where you're just using this sort of local corrective potential and applying those forces to an unfolded chain and seeing what happens, right? So the fact that the end states of the noising process are not unfolded proteins doesn't really factor in there. As the second thing you were saying about cooperativity, I mean, I think that something can be based largely on local interactions and still be cooperative, right? Because, cooperativity can be a function of geometry. For instance, if multiple local interactions together create a geometry that facilitates more local interactions, that's cooperativity, even though, it's driven in the purest sense by like range limited interactions, right? Yeah. Katie?
This could be a bit naive question, but I was wondering, when you're like, between the noise and like finding the energy from that,
Yeah.
are you able to direct that denoising towards specific energy levels and then like model across that energy level? Like it's like low energy, so it's still an actual structure, but it's high energy relative to the lowest state.
Oh yeah, sure, you could do that. Yeah, actually, I've kind of yeah, I mean, you mean like if you want to on purpose look at sort of like excited states or something like that. Yeah. No, you definitely can do that. There's a number of ways you could try that. But yeah, you could just try and like look at what the energy of the native state is and like literally just try to denoise to some, like you could like literally one nice thing I like about this framework is that you can optimize with respect to the model energy, but you can also add auxiliary like things that are going to direct the optimization too. So you could also just try to peg the energy to some higher level and see what happens. I haven't done that. I've tried to design things that are at particular energy offsets, like for instance, for trying to design some of these metamorphic proteins. And that does work. So yeah, that's a good, interesting idea. Yeah. Yeah, Nick.
Maybe I'll take the last question. So you motivated, one of the motivations
Yeah.
at the beginning of your talk was that as from a protein design point of view, one of the slowest steps is predicting your structure with AlphaFold.
Yeah.
And then filtering based on AlphaFold metrics.
Yeah.
And I'm wondering I don't know if you talked about it, but I'm wondering if you tried, like, take an input backbone, design 1,000 sequences with ProteinEBM, and then score them with your energy function.
Yeah.
And then predict their structures with AlphaFold and see if it correlates.
Yeah. I have tried that and it correlates very well. Yeah.
So should we all be using should we not be using AlphaFold?
You could try. I mean, I don't want to, if your protein design campaign is hinging on this, I don't want to make any too forceful recommendations. But in the test that I've made, and it's in the supplement of the paper, this quick test that we ran with just seeing like, how well does the energy correlate with like the refoldability score. And it correlates really well on the things we tested. So
It's much faster.
yeah.
Let's say change again. Right. Next month, we're going to be at Dana-Farber, and Jeffrey is going to be giving the talk. So hopefully you can come to Longwood Center then. All right.